Today, we are going to talk about an innovative use of infrared spectroscopy to screen potential cancer patients. We call this type of technique early cancer detection. In this video, we are going to explain what is an infrared spectroscope, how it works, and why it can be utilized to detect cancer earlier by simply measuring a human blood sample. First, let's check how early detection of cancer using blood testing is achieved. We know cancer cells multiply and spread indefinitely in the body, as well as releasing a variety of abnormal substances, such as metalloproteinases and collagenases. These abnormal substances can destroy surrounding tissues and basement membranes. After the barrier is destroyed, cancer cells will penetrate through blood vessels and lymphatic vessel walls, entering surrounding tissues and lymph nodes, forming metastatic lesions. To fight against cancer, our body also produces some other substances, such as interferon, which is a type of protein molecule that can stimulate the immune system, enhance the body's ability to attack cancer cells, and inhibit the growth and spread of cancer cells. We can thus identify the presence of cancer by detecting these substances in our circulating systems. Currently, there are two methods to test them in human blood. The first method is to detect the gene of cancer cells. The second method is to detect abnormal substances and other substances. Let's see how the first method works. We know when tumor cells die or divide, they release DNA fragments into the blood, which have mutations or variations consistent with the tumor cells, and are called ctDNA. In addition, due to the rapid division and growth, the cancer cells require a large amount of nutrients and oxygen, and because their cell structure is abnormal, making them more susceptible to various environmental factors. Therefore, compared to normal cells, cancer cells have a higher rate of apoptosis and necrosis. When these changes happen, they release a large amount of DNA fragments, which also contain genetic information of cancer cells known as cfDNA. Both types of DNA fragments come from the tumor genome, flow continuously in the human blood circulation system, and carry information such as gene mutations, copy number changes, and methylation related to cancer cells or other relevant cells. Now, we can use the method of cell biopsy to extract and quantify ctDNA and cfDNA in our blood. This will help determine the presence of cancer cells, the organ of cancer origin, and the progression of the cancer. However, the current data shows that this type of method has a low accuracy rate, with only a 75% detection rate of ctDNA in advanced cancer. Additionally, not all cancer cells release gene information, especially in the early stages of cancer, which makes it very hard to detect cancer earlier. So, let's talk about the second method, in which we can detect abnormal substances and other substances in human blood, which broadly fall into 7 categories: embryonic antigen, glycogen, hormones, proteins, enzymes, receptors, and genes. These 7 categories can reflect the occurrence of most tumors. Let's use one example to explain how this works. We know many proteins are only secreted during the embryonic period and stop being produced after birth. When tumors occur, the tumor cells will resynthesize and secrete embryonic-specific proteins, which we call embryonic antigens. This type of biomarkers include alpha-fetoprotein for primary liver cancer and carcinoembryonic antigen for common digestive system tumors and others. For example, when normal cells transform into malignant cells, the glycoproteins on the surface of the cells mutate and form special antigens, which are called glycogens. This type of biomarkers include CA50 for pancreatic, colon, and rectal cancer, as well as CA125 for ovarian epithelial cancer. So, how are these substances detected? Traditional tumor screening uses enzyme-linked immunoabsorbent assays called ELISAs, or chemiluminescent immunoassays called CLIA. ELISA can only detect a single indicator in one test. It is slow and costly. Therefore, it has gradually been replaced by CLIA. CLIA takes 2 hours to determine biomarkers and costs $100 to $300 per time. The challenge for these two traditional methods is that they can only test one different type of substance or DNA at a time. During the early stages of cancer screening, we don't have any idea if a person may potentially have which cancer. You have to run a lot of tests to find out if this person has a cancer. So, can we test all of the cancers at the same time? Yes, we can do this with a new technology called infrared spectroscopy examination. And the most advantage of using infrared spectroscopy is that it is not limited to specific cancer targets, as it can detect different types of tumor cells and different types of substances produced by tumors in the blood at the same time. Yes, at the same time. It's an amazing technology, and let's talk about how it works and how it is used to detect cancers. First, we have to understand the principle of infrared spectroscopy. Infrared is a type of electromagnetic radiation. According to its wavelength, there are also radio waves, microwaves, infrared, visible light, ultraviolet, X-rays, and gamma rays. The radio waves has the longest wavelength and the gamma ray has the shortest wavelength. So infrared has longer wavelengths than visible light and shorter wavelengths than microwaves. It is commonly referred to as heat radiation because it is mostly emitted by warm objects and can be detected as heat by the human body. Infrared is used in a variety of applications, including thermal imaging, remote sensing, and communication. Infrared spectroscopy is used to analyze the structure and properties of chemical compounds. And how it works? Infrared passes through a crystal with a high refractive index and is then projected onto the surface of measured materials. Here is the measured material. In this video, we call them samples. Refractive index is a measurement parameter of how much a material can refract light as it passes through it. The refractive index of a material depends on its composition, density, and other physical properties. The higher the refractive index of a material, the more it can bend light as it passes through it. For example, diamond has a very high refractive index, which is why it is so sparkly and reflective. On the other hand, air has a refractive index very close to 1, meaning that it doesn't significantly alter the path of light passing through it. Refractive index is an important concept in optics and is used to understand the behavior of light as it passes through lenses, prisms, and other optical components. In our case, not all of the infrared light is reflected back, but rather it penetrates into the surface of the sample to a certain depth before returning to the surface. During this process, the sample absorbs infrared light, resulting in a reduction in the intensity of the reflected light. The scale of the reduction depends on the structure of the sample. For example, if the sample is water, we use water to explain how it works, because water has a very simple structure and we know it well, so with the infrared spectroscopy we get a very simple wave pattern. There are 2 peaks that reflect the structure of water molecules. Let's use another example. Let's change the water to diamond as a sample, because diamond also has a very simple structure. With the infrared spectroscopy, we get a light absorption curve, or we call it a spectrum. With these 2 examples, we understand that when light is passed to the sample, the absorption light spectrum has different patterns for different molecular structures in sample material. Now, let's look at a spectrum of sample contains cancer gene or substance we mentioned earlier. This is a comparison chart between brain cancer patients and non-brain cancer patients. This is a comparison chart between breast cancer patients and non-breast cancer patients. And this is a comparison chart between colorectal cancer patients and non-colorectal cancer patients. This is a chart that compares kidney cancer patients with non-kidney cancer patients. The entire sample's characteristics, including cell-free tumor markers, tumor methylation markers, exosomes, proteins, and carbohydrate metabolites, are reflected in these spectrums. Based on this complete spectrum, and by comparing it with an existing database, artificial intelligence algorithms can provide results within minutes and show the possibility of the user having cancer. As you can see, it can be difficult to make judgments with the naked eye. That's why Discover also incorporates artificial intelligence algorithms to improve accuracy. The advantage of infrared spectroscopy is that it only requires a small amount of human serum, 9 microliters, which is dried into a powdered state on a slide. The instrument has many advantages, including 1. Simple and non-destructive sample preparation with no special requirements for sample size, shape, or water content. 2. It can perform in situ testing and real-time tracking. 3. High sensitivity and small measurement area with detection points as small as a few microns. 4. It can obtain the structural information of the molecules of a substance at the measurement location, as well as the infrared spectrum image of the spatial distribution of a certain compound or functional group in the micro area, which can be visualized through microscopy. 5. It can conduct infrared spectroscopy database retrieval and chemical functional group assisted analysis to determine the type and properties of a substance. 6. The instrument is relatively low priced and easy to operate. Now that we understand the process, it is clear that infrared spectroscope screening potential cancer patients is standing on the shoulder of giants. Their work involves refining the spectral library of different types of cancer. Once a patient's blood sample is analyzed with infrared spectroscopy, it can be compared to the database to obtain the desired conclusion. Discover is a cancer liquid biopsy platform developed by Discover Company based on this technology. It can predict the patient's condition by combining infrared spectroscopy with machine learning algorithms.